and then you can begin.
Okay. Awesome. Well, yeah, thanks so much. It's glad to be, even though it's the last session, but glad to be able to still present my stuff. So yeah, I'm Peter Searle. I'm a graduate student at Cornell University in the Gordon Lab. And today I'm going to talk about opsin gene evolution in an adaptive radiation of marine fishes. So the diversity of visual systems across the tree of life is really quite astounding, as you can see from all these different eyes. Visual systems are critical for survival because they allow organisms to detect predators, identify mates, and locate food. So understanding the visual system is really important and informative for understanding how species evolve. So organisms are faced with a variety of heterogeneous light environments. These include changes associated with light availability during day and night, as well as changes in turbidity and water depth, and really there's a variety of different things. Shifts in light environment changes what light is available to species, which has real implications for how species see and perceive their environment. Thus, visual systems really have to be able to respond to differences in these light environments. So there's several different ways that visual systems can respond to these differences, but today what I'd like to talk about is visual pigments or opsins. So these are essentially responsible for taking a photon and turning it into a physiological response. In vertebrates, there are 5 ancestral opsin classes. You can see here there's 1 rod and then there's 4 cones. Um, Rods are for dim light conditions, whereas cones operate in bright light conditions typically and are responsible for color vision. And then each of these opsin classes has a different maximum spectral absorbance, which you can see here. So opsins are really informative for understanding adaptive evolution because they can change via several mechanisms in response to changes in light environments. So these mechanisms include copy number variation. So if we were to look across the tree of life of fish, or I mean of all vertebrates or whatever, the size of the opsin repertoire varies substantially. You also can have different chromophore usage. So chromophores bind to opsin proteins, and depending on which one's present, impacts visual pigment sensitivity. You can have differential opsin expression, which also alters visual sensitivities depending on which gene's turned on or off or the level at which it's going. And then you can also have opsin sequence variation. So changes in specific amino acids can actually shift that peak in terms of what the protein can absorb. So in addition, these changes don't necessarily occur on the same evolutionary timescale. So variation at the copy level is often not as rapid as changes in gene expression. And now, although opsin genes have been studied really extensively in adaptive radiations of freshwater fish, there's a lot fewer studies that have looked in marine systems, despite there being plenty of adaptive radiations existing in marine systems. And so for this research, I'm planning on kind of exploring that area further in a group of fish known as rockfish, which is a diverse genus of marine fishes. And I'll talk a little bit more about these in just a second. So today, obviously there's lots of different levels that I could focus on, but I've just been started on this copy number variation. So that's what I'm going to focus on. And then the plan moving forward is to eventually look at some of these other categories. So previous work has found that copy numbers of certain opsins are correlated with depth across the tree of life. And it's been hypothesized that these changes are in response to the different wavelengths of light available in different light environments. So here in this figure, you can see that in the open ocean, the light that penetrates the deepest is in this blue spectrum, whereas in coastal waters, which can be productive and thus turbid, the spectrum can actually be shifted into the green, yellow, and often even into the red spectrum. So there's definitely some variability there. Previous research that's looked at this is consistent with these general trends. So it was found that in deeper water species, there were increased copies of this RH2 opsin. So I've color coded these for me to keep track and for everyone else to keep track. So RH2 is for the green spectrum. So there's increased copies of those. And then a decrease in copies of LWS. So that detects light in the yellow to orange spectrum in deep sea fishes specifically. Okay, so rockfish, it's this awesome rapid radiation of marine fishes, but that by my count, there's at least 112 species, but probably more. Rockfish evolved anywhere from like 9 to 8 million years ago. They started up here by this star in the Northwest Pacific Ocean. And then you can see in this map that they underwent several different rapid expansions. And we had some that got into the North Atlantic and even some that made it into the South Atlantic. So they've spread throughout a bunch of different areas. So rockfish are found in a variety of different habitats. So you can find them anywhere from like 0 meters in intertidal regions all the way down to 1,500 meters on the continental slope, and then anywhere in between. So this means that rockfish really, they can be found in the euphotic zone where there is light present, and potentially all the different wavelengths of light present. But then they can also be found down into the aphotic zone where there is no ambient light, or essentially very little light except for like bioluminescence. Um, you will see that the depth range of these species is quite variable. When larvae are released after the fish mate, the larvae actually undergo this like pelagic, it's like a temporary phase where the fish will go up into the water column. And then after a while, depends on the species, they'll often settle back down to the seafloor. So it means rockfish can be found in a variety of different depth environments throughout their lifetimes. Additionally, although few genomes have been evaluated for differences in gene copies, there is evidence that variation exists. So specifically in LWS and RH2, which are the genes that I'm going to talk about more. Now, you'll notice that these changes are small. Instead of seeing like multiple copies, it's like LWS is 1 or 0, and then RH2 is 3 or 4. It still suggests that even within a genus, you can have gene loss and gains happen pretty readily. And really, so all of these factors taken together make rockfish an ideal system to explore how changes in light environment could lead to adaptive divergence in visual pigments. So my hypotheses and predictions. I hypothesize that opsin genes have evolved rapidly in rockfish in response to changes in light environment, and predict that LWS, which again is associated with the yellow to orange spectrum, will be lost in deep water species, whereas RH2 will be retained or duplicated in deep water species. And I haven't really talked a lot about these other opsins, but previous studies have shown that SWS1, which is associated with the ultraviolet to violet spectrum, is typically less prevalent in deep water species, which would make sense because we're not getting ultraviolet light down that far. And then the opposite. So SWS2 is more prevalent as it is sensitive to the violet/blue spectrum. However, as the two rockfish represented here are actually really distantly related, if we were to pull up a phylogeny, they're like very separate from each other. It really suggests that there might not be any variation in these genes. Hence my prediction that there's no variation. And then for RH1, it was recently shown that RH1, so the rod, can show variation in like very, very deep water species, but it kind of seems like this is the exception to the rule. So for now, I don't really expect there to be any variation in that gene as well. So the first thing I did was I wanted to just be as thorough as I could and figure out how many rockfish species there were out there. And so I did a really thorough search through several different databases, and I identified 112 accepted species. And then I went on the hunt. I wanted to see how many, I want to do genomes. So I'm trying to get as many of the genomes, or sorry, as many samples as possible to assemble genomes. So luckily, there were already 80 samples on NCBI. And then with the help of a bunch of different awesome museums and organizations, I was able to acquire tissues from 27 other species. So that gets me to 107 of the 112 species. So far, I have sequenced just 11 of those samples. I'm still working on getting some more data back for the other samples. But these are data from some initial assemblies that I did. So I assembled contigs using this program called Mazurka, and then scaffolded the contigs with ragtag. And then a program that a lot of people use to assess completeness of genomes is called Busco. And so I use that program. It really is just like comparing what you have versus like a list of genes that you would be expect to find in any genome. And so what I found is that overall, many of these genomes are high quality, with the majority having 90% or more of these single copy Buscos. And then I use the orthologous genes from Busco to infer a maximum likelihood phylogeny using IQ tree 2. And here's the initial phylogeny. I really just kind of simplified it down because the main thing that I wanted to show was that it appears that many of the relationships within this phylogeny are well supported. So on the left, you can see the different colors. Red means less support for a relationship, whereas blue indicates a stronger support. And you can see that many of these relationships, they're blue, so they seem to be pretty strongly supported. Although I need to run Bayesian analyses and multiple other analyses to confirm that before I can really say that with certainty. So for an initial look of copy number variation, what I decided to do was just take the genes that were already out there and blast them using NCBI's blast tool. And if I found a hit, then I took that as meaning, well, with like a high percent identity, then I took that to mean that the genes were present. And then if there wasn't a hit, that was indicating that the genes weren't present. Now, this is a good strategy for like a first look, but it's not great for like finding duplicate genes. What I plan on doing in the future is actually using a different program called Minimap, but I won't talk too much on that so I can get to the cool, exciting results. So if we kind of put this stuff all together, what you can see is here on the left, we have a phylogeny with average depth mapped as a continuous trait. So red indicates shallow water species, whereas blue indicates deeper water species. And then on the right are the numbers of copies of both LWS and RH2. So I only got to these first two, but those were the ones that I was most interested in looking at initially. And then if a gene's missing, I've put in an X there. So in terms of LWS, I found that there were only 2 species, which you can see down here. So these are deep water species. They have lost LWS, but all the other rockfish species examined had a single copy of LWS regardless of the depth. So obviously this isn't quite what I was predicting. I think there's a couple reasons that this could be the case. One potential explanation is that many of these species are brightly colored. So the retention of LWS could be indicative of visual signaling in these brightly colored species. And then another explanation is that the pelagic shallow water phase in the larval and juvenile rockfish could be an important driver maintaining that copy number. So these fish are going up and coming down. And then in terms of RH2, there is one shallow water clade, which you can see here, in which most of the species only have 3 copies of RH2. And then if we kind of look throughout, there's also some other clades kind of scattered throughout. But again, in this case, there's just as many examples of shallow water species that have all 4 of their copies. So like here, this clade is clearly shallow water, but they still have 4 copies. So obviously there's lots more things that I need to do moving forward. So just in conclusion, understanding visual systems, particularly visual pigments, is critical for understanding how species adapt to changes in light environment. Based off previous work, I predicted that these deep water species are going to have fewer LWS genes and greater RH2 genes. Doesn't seem like there's much going on with LWS, and it does look like there's something going on with RH2, but I'm going to need to evaluate that further. And then really understanding opsin evolution in these fish is going to help understand opsin evolution in other rapid radiations. And so, yeah, just want to thank everybody that has helped make this possible. And if I have more time, I'm open to questions.
And you do, in fact, have time for questions. All right, so
Cool.
anyone want to raise their hand and we can then ask you to unmute and have you ask your question. If you are more comfortable doing so in text, you can also write your question into the Q&A feature if that would be easier for you. Okay, Parker, go ahead.
Hey, great talk. I just kind of had a question. So you were kind of just looking broadly at um, almost like opsin gene presence, but do you think that there might be a difference in like the gene expression itself that could be ecologically relevant as like a next step, or if you have predictions for that?
Yeah, so what I really want to do is pull the genes, then look at the sequence variation, and then the last step. And I don't know if I would do this across the phylogeny or if I would do it with like within like very specific species, but then would be to look at the opsin expression. Because other studies have shown that the fish might have those copies, but then functionally it doesn't even seem like sometimes they're using it. So I kind of think that like hitting each of those levels is going to be really informative.
All right, I am going to point out, actually, our second speaker is currently not in the room. So if there are any other questions, we do have time for additional questions. Otherwise, we are going to hold off a little bit so that way people who are moving between sessions don't end up coming over for a later speaker and having that person already having completed this. So any more questions for Peter? All right, well, in that case, I'm going to pause the recording.